Yeah, so to get, I mean, one of the biggest answers to how do we make Mike Snyder's world our world is money. Like you just, if you have enough money, you could do it today, of course. But that's true for many things, of course. We could solve it. Food distribution is not a problem of enough food. It's not about distribution of food or even money or resources. But then we get into questions of like communism, socialism, capitalism. That's a different class. But in any case. Money could solve that problem, but so even if you had all the money in the world, though, as a transition, what would you do if you had single molecule sequencing and whole genomes for every single person that is in the US or around the world? What are some good things or bad things that could come with that? So one of the last lectures is a bit more philosophical, a bit about the ethics, a bit about the interpretation, usability of the data, and even some designer genomes, and also a bit of intrigue about things you can do with data. Like if you could walk into a room and start sequencing everything on the surface, which you can do today, what would you find? What could you find using some of these like nanopore type sequencing devices? Let me close this guy here. Okay, so oh wait, you can see. So sometimes, so if you get your whole genome sequence, you get congratulations, you get your single molecule sequence data, you get a nanopore or PacBio genome, and you've got the beautiful genome, diploid resolved, you've got everything you want. And you might find that sometimes you could win the genetic lottery. So, for example, you could be a homozygous null mutant for myostatin and get these really large muscles. This is like a 2.5, 3-year-old kid who had these giant muscles and was lifting big weights. This is what the mutation looks like if you're a dog. So it's a orthologous gene looks like this. So exciting. You kind of get like little superhero powers as a dog. You could, for example, also get strong bones if you have LRP5 mutations, even a heterozygous. You can have really, really strong bones and become like the superhero who had unbreakable bones in this movie. Did anyone see this movie? Is anyone? I saw a few people. I found that as I get older, my cultural references become less relevant, so I need to update my slides. Like this used to be an interesting movie that people knew of, but in any case, now it's a good movie though. You should watch it. There's a trilogy technically, but so you could have really unbreakable bones. You could also, for example, have a mutation. Again, you could be kind of like a superhero here. You could also have a mutation, for example, though, that's on some of your T cells that could have a mutation in CCR5 or a chemokine receptor type 5. Which if you have mutations in this gene, makes it so almost every strain of HIV just cannot get into your T cells or CD4 cells, and thus cannot lead to AIDS and make you HIV positive. So kind of like getting a superpower, at least in terms of resistance to infection. And it's interesting about this is this was discovered really mostly on accident because of this paper. There was a long-term control of HIV discovered from a transplant where this patient had this 32 base pair deletion in the CCR5 allele. And this was, a good example where if you just monitor people, especially if you monitor transplants, sometimes you'll find like, that's odd. And like then you look at sequence the genome and you'll find, oh, this was why. It was actually turned out to be this mutation and that has since been discovered in other patients. It's rare, it's relatively rare, but if you have it, you have this kind of native HIV immunity. And I'll come back to this later with an interesting anecdote later in the lecture. But remember CCR5. And so there's some good examples where if you will, win the genetic lottery, but then sometimes you could lose. You could have high risk alleles. You could have alleles that give you, potentially, aortic aneurysm, or what was it, the aneurysm that Mike was referring to. My favorite quote from his lecture was, "The way you know you're at risk is if your aorta bursts and then you die for abdominal aortic aneurysm risk, which is not great, but hilarious. In any case, you could also have genes for hypertrophic cardiomyopathy, which is also relatively rare, but it's the most common cause of sudden death in young athletes. It can lead to left ventricular hypertrophy and outflow obstruction. And there's been dozens of different genes discovered, and hundreds at this point, actually not thousands of mutations have been identified. And so this is, one example where this was discovered in Reggie Lewis, for example, who had unfortunately died of this on the court. And so this was a case where people got concerned, and this is in the late 90s and the early 2000s, the early noughties, I like to call them, although popularizing like the not and the noughties. That hasn't caught on yet, but I think we should start calling the early noughties and see if that catches on. But so far it has not caught on. But in the early 2000s, this was a big concern. But genome sequencing was just getting started in the early noughties, right? It was just beginning. It was just kind of getting going. We didn't really have a whole genome reference until 2001, if you remember from the first lecture. It wasn't cheap enough to do. And so this was kind of an interesting phase of genetics in society where people knew about genetic risk, but they didn't have any easy or cheap ways to really test it. So it was something we could think about, but couldn't really test that readily. And so, for example, Eddie Curry was a victim of some of this sort of vagueness of genetics and society where, he was doing well. He was leading the team in scoring, playing for the Bulls, but then he had an irregular heartbeat and he missed the last 13 games of the regular season and the entire playoffs. And then, heart specialists and some cardiologists. Okay, seems like he's okay to resume practice. But then there was concern and said, well, does he have any of these risk genes for hypertrophic cardiomyopathy? Maybe he does, right? And so I thought, well, the general manager said, I understand the privacy issues, but I don't want to have something like what happened with Reggie Lewis, like I told you about, or other stars who died on the court. And of course, they're investing money and time in the players, and they're worried that they work for their own safety, but for the safety of the team, frankly. And so what's amazing is then they said, listen, the team offered an annuity of $400,000 a year for 50 years if you failed the genetic test. Like if you took a test but had their genes for risk alleles, they'd say, well, you failed, but you don't, we'll just give you this money every year, which is a pretty good deal. I feel like if you want to find my any mutation I have and say I'll give you 400K a year for 50 years, that's not too bad. But even still, he refused the test back in 2005 and said, no, it's not my genetics. You don't have the right to just, I don't know why, but it feels wrong, and I refuse to take the test. Like I don't want to know, because his salary was bigger than that, and he thought he had a bigger career ahead of him. But then he was traded to the Knicks, then he traded to the Timberwolves, and then the Heat and the Mavericks and the Golden Bulls, and ended up playing in China for a while. And so he didn't technically get fired for his refusal, but kind of got sidelined and then got traded and effectively was discriminated against. But it was technically not illegal for the team, his employer, to ask him for his genetic results. So this is just one example. But there's another example that was even a bit more intriguingly nefarious, which is one that is a famous case for genetic discrimination, where the Northern and Santa Fe Railway, you can see where they go across the country. They actually had something happening in the early noughties as well. Again, it's catching on. I can feel it. It's happening right now. It's catching on. But they had carpal tunnel syndrome, and most of you kind of know this because I think all of us work at desks, and you think, okay, I should get a nice desk that's got the right height and make sure I don't get carpal tunnel syndrome. We actually get this transverse carpal ligament. It's actually a lot of neurosurgeons love doing the surgery because it almost always cures the problem. Or you should also change your desk sitting. But anyway, at the company, basically at BNSF, they started noticing they were getting more and more cases, people basically applying for coverage and saying, I need to take time off, I'm getting carpal tunnel syndrome. And then someone at the company thought, well, aren't there genetic risk factors for carpal tunnel syndrome? For example, this one, if you've never been to OMIM, it's the Mendelian Inheritance in Man database. It's technically the online Mendelian inheritance in people, but they haven't updated the name yet, and OMIM is kind of catchy. But they, is the database of every known gene and linked to a specific disease. And here someone said, well, actually, let's just say we could start asking people. There's a summary of it and said, let's ask our employees to see, are they carrying, can you provide blood samples? Because yeah, you're filing for some disability for carpal tunnel, but maybe you were going to get it anyway, and then we don't have to pay for anything. It was basically what the company was claiming they could do. They then got sued. And then here too, they got sued, but there was technically no law being broken. So this actually ended up getting settled to the Equal Opportunity Employment Commission, rather than, because there's no other specific law they were breaking, and they got to pay the Americans with Disabilities Act by testing or seeking to test 36 of their employees. And it's interesting, it seems like, well, maybe there's a change that's needed. So clearly, people are just asking people for the genetic results. And this led to actually eventually in 2008, the President Bush signing the Genetic Information Non-Discrimination Act. So it makes it illegal to hire and fire or change essentially, health insurance based on your genetics. Because the idea is it's something, obviously, it's an immutable characteristic. You're born with it. You can't just change your DNA, at least not yet, as easily. I'll get to that at the end of the lecture. And the idea is we should be protected against this. And so it protects, for example, against health insurance. So you can't just say, I'm not going to give you health insurance because I saw your sister posted on Facebook that you said you had BRCA, or that you posted that you're BRCA positive. So you can't, or if you can't ask for testing. So this is for health insurance, for whether you get it or not, the coverage, underwriting, or premiums, and for employment. So for hiring, firing, promotions, you can't just say, well, you're BRCA positive, I'm just not going to hire you because I'm worried somewhere down the line we might have more health insurance costs. So what's interesting about this, though, is it doesn't apply to smaller companies. If you have less than 15 employees, the idea is that it's too complicated for small companies. This seems really weird, but it shows up in a lot of legislation where if it's a really small business, the idea is, well, it's too hard for a small business, which I think is kind of crazy, but apparently this is more common. It doesn't apply to the military or the Indian Health Service or the VA. It also does not cover long-term care or life insurance or disability insurance. And so what's interesting, though, is other people have been thinking about this. Florida just in 2020 expanded it to protect discrimination from life and disability insurers. And so people are thinking about expanding it to beyond just health insurance, going to life and disability. Which is important if you think you might be at risk for ALS. Like say you have some of the genes here. If you have some of the genes we just heard about from Snyder, and if you have ALS, which actually I just had recently a family member diagnosed with ALS, I'm sure he wished he would have had some ability to expand life insurance or disability insurance, but they certainly cannot do it now. Now he's been diagnosed, but even if you're at risk to be able to do so. But of course, that changes the moral hazard. It changes the calculation of the insurance agency, so it is more complicated than just expanding the law. But I want to tell you about a few applications. So one of them is one of the first uses of GINA was the case of the devious defecator. Have you ever heard of this story? This was a fabulous case where basically a mystery employee began habitually defecating in one of the warehouses for this Atlas Retail Services Group. And what's interesting, you think like, well, that seems really odd. And then you read about some of the articles about this, it turns out it's not as rare as one might imagine. There's several cases where people start defecating in warehouses, which you wouldn't necessarily imagine happening, but apparently it does. And so the employer said, okay, I think I know who it is, and I'm going to ask these two guys and say, okay, I think it's you guys, and I need a sample. I think it's you because I can look for the human DNA in the stool and prove that it's you. And they said, well, you can't do that. That's not legal to do. And eventually they did. They sued after they were ordered to take a test, and then they won. And it was kind of the first case of the devious defecator case, landmark for U.S. genetic privacy. Again, this is a kind of colorful way to display the applications of it, but it was true. They say, you ask me for the sample, you're not allowed to, and so they actually won the case. And some of the damages were punitive as well as actually for corrective. And so, and this is not just for human DNA. You could imagine a world where you ever see dog poop on the sidewalk in New York City, right? There, it's everywhere. But if in theory we could do is just get it so you could take every time you register a dog, you keep a piece of the DNA. You could sequence it right on site and say, okay, we know which dog this is, and you send a ticket to the owner, include the cost of the prep and sequencing as part of the cost. And in theory, then you could actually recoup all the costs. And this has been discussed in some places as well. And the reason I want to bring this up is because all of the DNA that is a sign that came with one of the campaigns, is all the DNA that you leave behind tells a story. So I want to think the idea of this idea of sequencing anything anywhere at any time then begs the question of, well, what could someone learn about me, and what am I putting at risk basically by just like leaving this room? Like when you get up out of the chair, what are you leaving behind? And so I'm going to tell you a little fun, many people think terrifying story of everything that we could learn. So in particular, all of us, of course, look different. We have the shared ancestry of humans that likely moved out of Africa about 100 to 150,000 years ago, moved across the world and essentially populated the world. But this local selection pressure and population bottlenecks has led to why we have different hair color, eye color, skin color, we have different languages to some degree. So this really interesting dispersal of humans across the planet has given us little pockets of genotypes that were slightly different. So for example, if you look in Europe, one of the first papers by John November that just said, okay, let's take genetic data from across Europe and see, can we remake the map of Europe just from genetics data alone on a principal component map? And there you basically kind of can. It's not perfect, but you can just take by the distance, the genetic distance between populations, what they look like. And you can even see that within a few hundred kilometers where someone's likely born based on another figure from this paper. So you can start to use the genetics to guess your ancestry. Now, it's not perfect, but you can actually get pretty close, again, within where you're likely from, say 95% accurate. And what's interesting about this is, we used this came up in the class earlier today, thinking about clusters across New York City. Nick, for your talk, but actually, we took census data from the United States. In one paper, we sequenced all the subway stations across New York City and tried to predict the ancestry based just on the subway station. So you can see here, the census data is Asian, Black, Hispanic, and white. They might expand this in the next census, but right now those are the 4 categories, which as a geneticist is just infuriating to say you have to lump everyone into these 1 of 4 categories because it's wildly oversimplified. But you can see here, we can reconstruct and recapitulate the likely ancestry of what's in Canarsie, for example, or Mount Eden or Chinatown. You can see bursts and changes of the ancestry of the subway stations left behind. So we could to some degree tell what subway station you're in. We could maybe try and predict what you look like. So this is actually done by a genetic artist named Heather Dewey Hargberg. We take DNA from cigarette butts and try and create the hair, eye, and skin color and make these kind of creepy looking masks of what you likely look like based on the DNA left behind. And so this is for ancestry prediction, hair color, eye color. It's not perfect, but you can get essentially a prediction of what you think someone might look like. And you're also an echo of your family. So of course, if you're an identical twin, you'll have almost 100% match of DNA, but your siblings, cousin. When I see my cousins, I often say, hey, 12.5% of my DNA is the same. And they're like, oh, you're so dorky, but it's a fun statistic. You should do it every family reunion and throw out numbers like this because I think it's fun because I'm a geneticist, and you think about your grandparents, of course, we're all related. What the FBI uses are these combined DNA index system to basically take short terminal repeats in your genome, which are all you can do by PCR is try and guess the size, but by long read sequencing, you could actually get the sequence of these short tandem repeats all across the genome. And so the FBI still use just primers here, and they added more loci recently. But they basically try and guess, is it you based on, and do you match the database? But based on this, if you're in the database, they'll find you. But say you have a sibling that commits a crime or a first cousin, that means you'd start to do matching. So they do what's called familial matching. And this was used to find someone who threw a brick off a bridge had killed someone. And they canvassed the neighborhood and found his brother who had committed a crime and said, "Oh, has your brother been out today?" And said, "Oh, actually, he was out by that bridge." And they found him just by familial testing. So you probably already know that it's not a good idea to commit crimes. But if you have family members who've already been arrested for committing crimes, you definitely shouldn't commit any crimes because they could find you through familial searching. And if it's worth knowing, of course, that it's probably, CODIS may be agnostic to its subject, but society is not. It's probably, there's a fair amount of bias in terms of people getting arrested are more likely to be Black, and then they just take DNA, and they might even get convicted, they'll just get arrested. So there's a lot of problems socially with how CODIS is deployed, but I want to point out what you can learn from DNA left behind. So don't commit any crimes. That's point number one. But what else can molecules reveal about you? Of course, you remember from the second lecture that DNA methylation is an echo of your life as well. So we talked about epigenetic age. And remember what happened the day when egg and sperm came together, when you had the Marvin Gaye and the candlelight and the wine, and when egg and sperm came together. And this is the rapid demethylation of the paternal genome on top, which then after you have division, and you slowly actually reset all the epigenetic marks. So we talked about this before, that there's epigenetic age, just as a quick reminder, that increases in all and across all tissues, and you can fit a training model and predict it, again, just as a quick review, and that this has been validated by other groups. So in theory, if you have a piece of DNA that's left behind in this room, I could try and tell how old you likely are just based on that DNA alone. And so this has been used actually for once at the scene of a crime. We did this for blood actually left in a car for the case of making a murder with Stephen Avery. That there was a question whether his blood 10 years old that had been older, or was it fresh blood? We had to distinguish between whether it was the fresh blood or the ancient blood. Turns out it was more fresh. And there's other companies that have come out that have started saying, well, do you want to do services to see how old you are, or would you like to see your biological age? Do you want to see if you have quality control for your reprogrammed cells that can tell you how reprogrammed they are? And so it's interesting. You can imagine a case where, it used to be hard to get a fake ID when you want to go to a bar as a kid, like you're 18, you want to go to a bar, you had to get like an older sibling. It was very complicated, but now you can go to YOLOIDs.com and get a fake ID from any state in the union if you want to. And I have no relationship with this company. I just think it's fascinating that this exists. Yes, you can pay by Bitcoin with this company if you want to. And you could imagine where you're going to get, you got the unit fake ID, but you go to a bar and someone's like, no, I've got a nanopore sequencer that's set up right at the front of the bar, and I can tell how old you are and whether you're going to get in or not. And now this has not yet happened, but I think in the future, to have the most expensive way to validate age, we could maybe do it as a concept bar in New York City, I bet, and people would pay $500 to enter. That would probably take off. But in any case, this has not yet happened, but it could happen. Certainly it's been happened for crime scenes, which we actually did nanopore sequencing and bisulfite sequencing, and someday maybe in the future bars. But this has led to some people to say, myself included, maybe we should update our non-discrimination laws because epigenetics is a factor that you could consider, or other microbiome features, really just about genetics, not epigenetics in the original legislation. And of course, there's a lot of companies now saying we can tell how old you are based on your epigenetic age. And so that's kind of that's the genetics, and the epigenetics is what you leave behind. Another thing that you can and do leave behind is your microbiome. So everyone here has a unique microbiome. This has actually been studied by many, many groups over the years. One of them, for example, is that even after an antibiotic treatment, some of your fecal microbiome or a fecal microbiome transplant, your individual signature will remain in your gut sometimes. So we all carry this unique signature that in theory, you could track someone. Like imagine if you had a nanopore sequencer attached to every toilet across New York City. And you might think, who would do such a thing? But I think it'd be a fascinating study. I would do that immediately. But if you could track where someone's going across the city and see map where they're going. This, of course, has raised questions of privacy. Or if you can track everyone, what does that mean? How do we protect against that? Should we have, basically protection for your gut prints? And you can even imagine if you just look around the city. I mentioned we do this work of looking at subways and urban microbiomes. Even each shoe, if you just took shoes and said, well, could I predict what city you came from? If you land on a plane, I don't ask you any questions. I just take your shoe and I swab it and sequence it. Could I tell? So one way we've been able to tell this is we have a project called MetaSub, is the metagenomics of subways and urban biomes, and we have a global city sampling day. You're all welcome to join on June 21st if you like, where we create a global microbial census each year to see what's really present in each of these cities as a way to map new organisms, track viruses, antimicrobial resistance, find new molecules. But there is also an interesting facet of this data that lets us actually track where you may have come from. It has a forensic feature where we can, given the number of samples, get to about an 85 or 90% accuracy of city of origin prediction. So just take any random sample and I can tell you where you're likely coming from in the world. because just like at the macroscopic level, there's different unique flora and fauna that are present in different cities and around the world. Same thing is true microbially. And so if you look in aggregate across all the things we could predict about you, and what your forensic profile is, your social profile, Amazon's tracking you, Facebook's tracking you, Twitter, you could start to just feel like, that you should just maybe give up because everyone's tracking you. I don't think you should give up, but I think you should know that everyone's tracking you and that there are molecular means by which you can ascertain where someone's likely been, what they're doing, how old they are, what they've eaten, and general lifestyle features. So again, most of these are probabilistic. They're not going to be used to solve crimes. They've been used a little bit epigenetics in some crimes, but they're not yet used by the FBA, not yet. But this might also start to raise questions in your head. Okay, is this legal? Can you patent these methods? How is this even possible? And so I want to switch gears a little bit on the policy side as well. Of like, well, what happens to the world we live in if this starts occurring more routinely? And so it depends on what you mean by expectation of privacy. So when you leave this room, or any of you on Zoom, when you leave your chair, it's harder for us to come into your house if you're at home because you're on your chair. But also, if anyone leaves this room, it's very easy. But if, for example, in your home, one of my favorite examples of expectation of privacy is this Supreme Court case, Danny Kylo versus the United States, is that he was an ambitious young man and wanted to grow some weed in his apartment, and it was using a lot of electricity. And then the police used thermal imaging cameras to notice that his window was really active in the daytime and night, and because he was having growing machines, which is now legal in New York City, but at the time it was not. And so, he said, "Well, this is not fair. It's not. I have a reasonable expectation of privacy that people don't have thermographic vision to see what's going on in my apartment." So he challenged his arrest, went all over the Supreme Court, and then he won because the justices said, "Yeah, that's true. You can't really. Most people don't see in that spectrum of light, right? That's true. So you do have a reasonable expectation of privacy then in 2001, but today you can get drones that have thermographic cameras on them just situated for like a few $100s. So I don't know if this case would be decided the same way today. Probably not. And this leads to a little bit of a sidebar for 10 minutes of what I did for some work when I was a postdoc, was just this question. So it's privacy question, but even just patenting, could you just take a gene, say I want to take a gene out of my body or from a surface and I sequence it, and for a little while people would say, no, that's my gene. As soon as you sequence it, it's my property. As soon as you isolate the DNA, I remember this is coming in the early noughties. I think it's catching on now, the noughties. And it's a bit of history that started actually at a place that was in New Haven, Connecticut, a place called The Anchor. And it started with the guys chatting with the bar called the Don, and we're having beers. Now, normally if you have beers on a night and you talk about something and, hey, we have an idea, it usually leads to nothing. But sometimes if you remember to write back the next morning of what you were talking about and take notes while you're chatting, it led to this idea of like, we should look at the intersections of law and essentially law and genomics and sort of law and order genomics unit, like Law and Order, the show, the bump show. Anyway, we tried to make it like this. And it did basically a course, got this set up. And so I was a postdoc looking for disease genes. I was taking patient DNA, sequencing it back then, even Sanger sequencing before we had single molecule sequencing, and was looking for whether we could find genes that might be associated with brain malformations or disease or autism. And I was in lab late one night and found this paper and said that apparently about 40% of genes had a lot of filing that might be, but 20% of genes were explicitly claimed in US IP. They're actually like they were patent, like the genes were patented. So that doesn't make sense. What do you mean the gene is patented? Like, well, as soon as you purify it out, it's no longer your property. You purify the DNA, or certainly if you amplify it. So I was really confused and thought that just doesn't seem, it just seems wrong. The same way I think, some of the genetic discrimination cases where people just thought, that just seems wrong. It seems like I shouldn't be able to do that. And so then around the same time, actually, there's a place in GenSpace. If you've not been here, plugs in here. Let's plug in there. It's fine. It's fine. A place in Brooklyn. It's actually called GenSpace. It's a community lab. So you might think, if you want to go join a gym, you pay a gym membership. But if you want to join a lab, you can pay at a GenSpace. And they have different events. They have events like, say you want to do a molecular gastronomy course, or let's say you would like to PCR your own DNA and say when you want to say, I'd like to take this course where you can have PCR and pizza, or you can CRISPR your own cells, for example. You can do GFP cloning. So one night I got this is a community support community labs, old picture from their website. And you could get a gift certificate for a 1-day personal genotyping workshop. This was before I had my own lab, so I had to do it this way. I guess that was coming up here. But so this is really cool. You can go there and clone some of your own genes. So I got some of this gift certificate for some friends because I'm a big nerd. And then said, okay, we can do a PCR of your specific loci and see, do you have this deletion? In particular, the CCR5 delta 32 deletion. So we could all basically see who has the genetic superpower. And basically it was kind of exciting. And then I had one friend of mine, Rob, who was just like, oh, if I have the superpower, I could go crazy tonight and like hit up and go get like, 5 girlfriends. And then I said, well, no, there's other diseases you can get, Rob. But anyway, he had this really interesting reaction. He didn't have this genetic superpower, so I guess he was safe that night. But yes, it's the same gene, the CCR5 gene. And so what's interesting about this is just taking out DNA from our own body and doing PCR on it was technically an infringing act, as written a lot by Steve Salzberg and others at the times, that if you purify innovative DNA that's patented and you test it at that time, it was an infringing act. So you're basically violating a patent. And so, but what that means also is that infringement for any modern day geneticist was unavoidable. I mean, I think just to remind you a bit of what patents are, is they have to be something that's new and useful and non-obvious. So it's really in the Constitution. So people say, where did this whole patent system come from? It's in the Constitution itself of our country, is to promote the progress of science and the useful arts. For limited times, you get to authors and inventors exclusive right to their respective writings and discoveries. Now, originally, now it's 20 years, and so you can actually have, as long as you satisfy these 3 criteria, you can actually then get a patent. And so what were some of the patents, for example, at that time on BRCA1, the cDNA, as well as the gene itself? It was any isolated DNA that has anything that was in the sequence of the patent, and then anything that the nucleotide sequence that was kind of related. Okay, the gene itself, the purified gene, but then any the other claims were anything that has at least 15 nucleotides of the gene or the cDNA. Which you think, well, 15 nucleotides, that seems kind of it seems kind of short, right? That seems like that would probably, if you do a sequence analysis, that would probably show up everywhere. And so indeed it did. I did the analysis and said, well, how many genes have at least a 15-mer that shows up in any other gene? So actually 100% of genes have at least one 15-mer that matches another gene that's in BRCA1. So we published this a few years ago and thought, well, this just seems insane. And that's actually just looking in the genes. If you think about what we're looking, that's in the exons. But there's all these other areas across the genome. There's transposable elements, if you remember from the first lecture, all these other areas of the genome, which we can now resolve by long read sequencing, much better, in some cases completely novel. And for example, there's a BRCA1 pseudogene in the genome that's just upstream. So a whole version of the copy of it's in cDNA form. And of course, it's important not just to think about the gene, but also the fact that there might be mutations in BRCA1 that are important for breast or ovarian cancer. But if you look at the CBio portal, these are all the other cancers and how often they're mutated in BRCA1. So it's not just breast and ovarian cancer. It's a DNA repair modulating enzyme. It's important for almost every cancer, right? So BRCA1, same with TP53 or NPM1. A lot of these genes are check inhibitors, PI3 kinase. A lot of genes are important for many cancers because BRCA1 has pleiotropy. One gene has many functions, as most of you know. So what we did is we then I filed briefs in 2009, 2010, and went all the way to the district court, the appellate court, went to the Supreme Court. I wrote op-eds in the Washington Post and saying they should invalidate the patent on human DNA. The CEO of Myriad Genetics, who was at the time holding the patents, and wrote back to the next day and said these guys are morons and they don't understand what they're talking about, which I think was very fun. So I'll probably never get a job at Myriad Genetics, but that's okay because I feel like we were fighting a good fight. Then partnered with the ACLUs to work on this case. They then filed on behalf of patients to say, I just want to get a second opinion. I'd like to sequence my genome at a second location, or at least my gene, which you couldn't do. And they said they had a little logo, do not patent my genes. And then there's all these op-eds coming out. I went to the, on April of 2013, went to the Supreme Court. And then you can see here, it's kind of right after the oral arguments came out, everyone's very excited. I saw Francis Collins high-fiving people outside, he was just the previous director of the NIH. And then after the challenge was made in 2013, actually a few months later, the claims were rejected. And so what's extraordinary about this is within one day, the cost of testing, a bunch of companies immediately released testing because it was much cheaper. It's shotgun sequencing for a whole genome is about this much, or even around that time, it wasn't too much more. But certainly for a targeted panel to sequence, it was about $250. And so it really was extraordinary to see this immediate impact on the market, on testing, on accessibility to DNA. So I was very excited to see that as a scientist, as a geneticist, as a person. And so it's worth noting, though, that was then, but the battle does still somewhat continue in 2022 and also just last year as well. There has been patent bills. You might think patent legislation sounds like maybe the most boring possible thing, and to most people it is. That's why it doesn't move quickly through Congress. But there is still a Patent Eligibility Restoration Act that sometimes gets introduced as bills over the past really 10 years now. So the battle still comes on. It could get attached to an appropriations bill. There is an ongoing battle for this question of can you patent your own? Can someone patent your genes within inside of your body? So far, still the answer is no. But I want to give a little bit of background. on those genes. And so in the last section of the talk, I want to think about, okay, well, those are the bad genes, but this all assumes in genetic discrimination, genetic sort of modification, all assumes that you are given a genetic shuffle at birth and that it's immutable, that you can, that it is what you have and that you can't change it. But of course, I think as you've learned in this course and probably already knew, is that it is not that simple anymore. You could actually do somatic modifications of the bad genes, depending if you really understand the bad. You can also do even germline potential modification. And so an idea, a dream would even be, I guess the Snyder dream is that we're all modifying everything, in response to wearables and health, but also another dream would be, I think that you have not just, predictive medicine before you get to have disease or when you're a modification of health. But when you do get sick, you have precision medicine, but they keep going back and forth to being predictive as to whether you're on the right trajectory or are you starting to become towards sickness or losing your health. And so there's multiple ways to look at this. You could look at, say, BRCA1 mutations, P53 mutations, cystic fibrosis mutations, hypertrophic cardiomyopathy, and say, okay, we could just repair them in the embryo stage. And so this was first hypothetical, but it became very, very concrete in 2015 when the first gene-edited embryos were published. And so this suddenly became no longer hypothetical. And then, of course, there was, additional modification. People talked about this. True, it reignited the debate of, well, should we even do any germline modification? The answer is, I'll come to that at the end. But there's very few cases where you could have a strong argument of yes, but maybe two. But then the additional editing was done, smashed in pigs, where you could do multiplex editing of 60 genes. Actually, there was a xenotransplantation of a pig kidney a few months ago. The patient unfortunately just died a few weeks ago. But shows that it's possible to actually reduce a lot of the porcine endogenous retroviruses that could create an immune response and then actually make it so you can do xenotransplantation or one species to another. Pigs, I think, is a weird ethical question. If you get a kidney transplanted from the pig and they have to sacrifice the pig, should you have pork chops and bacon from the pig that gave you its kidney? Is that cannibalism then? Are you eating yourself? What does that look like? Impolite to not have the pig, it just goes to waste. I feel like someone should do something with the pig if it was used. Is it safe to eat? There's a lot of questions we don't know. But in any case, there is ongoing work on xenotransplantation. But the case that most of you are probably thinking of in terms of editing babies or editing embryos and then implanting them and having them be born was the case of He Jiankui, who announced onto the world, it said, I've done this. It's happened and the baby's been born in 2018. And so it was no longer a hypothetical situation of could we modify embryos in germline engineering, germline meaning all the cells that could be passed on to the next generation. And of course, he did it in secret with kind of a sketchy IRB. I've written a lot of IRB protocols, and you have to be pretty explicit about what you're doing. He did say he was doing this work, but it wasn't as clear as to what it would mean, and it was vague about a lot of parts of it. But the interesting thing about this is, what's the gene that he chose? He actually chose a gene to think, okay, I want to try and pick a gene that, and I think in his mind would be unassailable. This is something that is a defect in the genome that if I correct it, it will lead to the patient safety. They'll be better off for it. What he chose was CCR5. So he chose this specific gene that would give you a native immunity to most forms of HIV, almost all forms of HIV. But actually, one of David Liu, who was in the audience when he announced it, it said correctly and posted a lot on Twitter at the time and said, what was the unmet medical need that was being addressed by doing this to actually do CCR5 modifications of embryos? And the answer was there isn't, right? You can actually avoid HIV just with condoms or just essentially with basic measures. Even if you get HIV, life expectancy is now on par with most people who don't have HIV. So the world has changed from what it was, say, 20, 25 years ago of an HIV diagnosis. So that's one reason why it's important, or multiple reasons. One is that it was risky. He also did a regular CRISPR that almost certainly had off-target effects. I'll get to that, other ways to improve it shortly. But it was the very early version of a very crude tool to address a non-needed medical issue in embryos that were otherwise fine. And the other thing about, I think CCR5 is, of course, it of course has pleiotropy. So CCR5, you're like, okay, you might limit your risk of HIV to near zero, but then you'll probably be at a higher risk of West Nile virus. So, getting rid of a receptor in one case helps with one virus, but might hurt with another. So the fact that it has pleiotropy, it says CCR5 save lives, some people say, for example, in this article, is that it has multiple functions. It's not just one function for one gene. Even though Beatum and Tatel did get a Nobel Prize for that, I think it's more complicated than that in mammalian genomes. So I think this is something that just have to remember. And so it leads to the case where now we're thinking about other modifications, and it's not just individuals, but there is a consortium of people. They're anchored actually at New York Genome Center and other sites of how do we write genomes from scratch. We could edit them as well as you could edit DNA as well as begin to write DNA. And there's also an NIH somatic cell genome editing program that's active, giving hundreds of millions of dollars towards these efforts. So this is no longer, again, no longer hypothetical, it's ongoing, but it's more for somatic editing where you're doing the cells in the body. And some people do still talk about ways to do, repairs of genes for, say, hypertrophic cardiomyopathy, for example. This is what Shukat Metelipov, but I also think it's a very funny picture. He looks like, he's very, he's happy. Is he happy? I can't tell right there, but he's, he definitely finished editing some embryos. I think it's the editing embryo phase. And so there's some ongoing work that's still happening. And it's not just in, modifying, for example, embryos or doing somatic editing. There is also a CRISPR therapy race that's being done for T cells. Actually, we've worked a bit with one of the clinical labs in China, where actually, if you look at where all the CAR T trials, the chimeric antigen receptor T cell therapies, most of them are actually in China. You can see in red here. This is a review we did in 2020. This is only exacerbated in the past few years. More of the clinical trials are in China. And they're not just for liquid cancers. You've probably heard of CAR T therapy. This just shows you where they're occurring, what phase of the trials and the recruitment levels. But they're now actually, you can see these are liquid cancers over here, and also solid tumors over here on the right. So both liquid and solid tumor modified T cells with CRISPR are being used therapeutically. So this isn't quite somatic CRISPRing, but it's just CRISPRing of T cells and then using them therapeutically and infusing them. So it's a kind of a version of somatic therapy. But my favorite example to date, actually, of thinking about creative ways to use CRISPR modifications was people started talking in 2018 about, should we actually do modifications for like targeting genes, for example, like BCL11A, and making these custom Cas enzyme edit adult hemoglobin. The reason would be, if you remember when you were a baby, again, remember during the Marvin Gaye and the Wine and the Candlelight, when you originally were a fetus, you did have fetal hemoglobin, which you can see here is sort of an epsilon and zeta version of hemoglobin. And eventually you kind of get through your life, you have beta and alpha hemoglobin, and that's mostly what carries oxygen around your body. But there's sometimes a little bit of delta that comes up. But for the most part, it goes away. But so what that means, though, is there actually is another version of a gene that makes hemoglobin that's already in your body that just is kind of silent. It's silenced essentially by an enhancer that's in front of the gene, the gene BCL11A. And so what's interesting is people started thinking about this back in 2017, 2018, saying, well, what if we just bring back, bring back gamma or bring back epsilon or zeta? Could we bring back something else that went away and then repair it that way? And so that's indeed exactly what happened 3 years ago. Was the first successes already showed for editing basically this gene here. You can see your BCL11A. And if you just delete this erythroid enhancer region, suddenly you can reactivate the gene. And then if you look here in blue, this is the fetal hemoglobin that's being populated several months after the infusion of the product. I have no relationship with this company. I just think it's one of the ideas like, well, let's turn back on a gene with a small deletion. But that's actually doing it more with genetic modifications. We talked in previous lectures about you could do it also epigenetically. Material, you could also turn it on, and you could do it stably. You could in principle do the same thing with BCL11A or other genes that could give you genes that you want inside you. There's actually a pseudogene for vitamin C production in our bodies. I would like to reactivate that, but that's for a different discussion. But in theory, anything that's in us, you could activate, or anything that's not in you, you could add back in. And so it just makes it a really extraordinary time. That also means that was with original CRISPR methods. David Liu and others have published even better methods, which are one's called prime editing, which doesn't create double strand breaks, because that can also enrich for cells that might be P53 negative. So if you can tolerate double strand breaks, what kind of cell are you? Probably not a good one. So you want to think about systems that only modify one strand and don't create double strand breaks. And this indeed, but prime editing is now used in a lot of clinical trials. Actually, now I'm thinking about doing this next few days. I want to see how many there are. We haven't looked recently, but I'm sure there's a lot more. And this is kind of one of the methods that's exciting. And as I mentioned also in the previous selection, you can also do epigenetic correction as well as the detection of what's wrong. And this was done in mice to do DNA methylation editing of the FMR1 gene to rescue Fragile X syndrome, for example. So some of these, again, are early studies done in mice, done in small cohorts clinically, but I think really open this brave new world where we can actually think about, create, and deploy some of these kinds of therapies. So in closing on the sort of thinking is, there's a good idea to upgrade our DNA. Of course, it becomes a big ethical question. And you see headlines that look like this, like, should we eliminate changes that would pass down? But should we do such a thing? And I think what's interesting is, there's other examples where things seemed odd, but then became normal. So for example, 3-parent babies, you could have, 2 parents, but then also mitochondrial donor to have 3-parent babies. That's become more common. There was the MPs voted to actually make it possible in the UK. And Jennifer Doudna mentioned that people get comfortable, and won the Nobel Prize, for CRISPR work, that people will eventually get comfortable with these new technologies, much like they did with IVF, which is now 1 to 2%, even as much as 3% of all births in vitro fertilization in a lot of industrialized nations. And so I think, to close on the thought, I think even though people get comfortable with it, there is always the thing that you should always ask is there an unmet medical need? If I'm trying, I mean, we can do all kinds of things in the lab with mice and with cell lines and do all kinds of things, but when it comes to deploying something in patients, is there an unmet medical need? And if the answer is no, or even if the answer is maybe, then you probably should just, do preclinical work, think about it, optimize it. But if you're not really addressing a need, then there's probably no need to do it, which is what He-Jin Kui, I think he really failed. He didn't actually have a solid yes for that answer, for that question. But there's two examples I alluded to before where it's possible germline editing may be not only possible, but even more ethical than not. So the only two cases would be, because if you think about this, what if somatic editing gets so good in the next 5 years that you would never need to do germline editing, because you can get as good as germline by doing complete somatic modifications of every cell in your body. So you wouldn't need to. And then you could do deep sequencing and confirm there's no off-target effects and say, okay, we've perfectly done it. But there's two cases maybe where you would need to, and that one would be where if you have a couple that say both are homozygous for two mutations and they don't physically have the alleles to create a healthy offspring, you could then modify their gametes though to make it so they do carry a wild type allele of some disease gene, like Huntington's disease, could be cystic fibrosis, but probably not a disease gene where it's a higher risk. Like BRCA1 gives you some of those mutations are 90% risk of breast cancer, but some are a 40% risk of breast cancer, lifetime risk. And so is that enough to modify it? Probably not. But if some cases you have a homozygous, two parents who fall in love and they want to have a child, and they can't genetically create one that doesn't carry the disease, I would want to enable them to say, okay, we can modify our gametes, so we could have that. I think it's one case maybe that we'd be ethically able to defend it. The second case is more peculiar, where say you're sending people to another planet with higher amounts of radiation, you want to activate and add a bunch of DNA repair genes so they can just survive. So if it is essential to survival and you don't have the tools and resources to do continual somatic editing, because it would be more expensive, you say, we're just going to send the humans to the new planet with sort of the genetic payload right out of the gate and hope that they survive. Otherwise, the alternative is just go there and be radiated and probably die. So I think in that case, you'd be ethically bound to give them all the genetic protections that you possibly could. So they're probably the only two cases where I think we could think about germline editing as maybe being ethically defendable, potentially. But there's also some questions I'll come to at the end. But that's actually, but that's it. So it's kind of the bookend of the course starts to get you thinking about the impacts on society and editing and sequencing and where we deploy it, and the fact that no one really has any privacy anymore. And that's both socially and genetically and molecularly. So don't commit any crimes, as is the other lesson for the day. And happy to take any questions. And I see some actually here. Hold on. Omar, okay. Oh, you can see it over here. One, there's just more mutations, increased rates of fertility, so there's more diversity and genetic selection. So could you basically, how do you prevent segregation towards people that opt out of such editing? There is I like to differentiate challenges, ethical questions into two parts. One is there's a tactical question and there's a principle question. So the tactics is how do we distribute technology? And so we already know quite well, there's disparities in the healthcare system. It's probably most worse in the United States, but in everywhere in the world, usually the rich get early or even exclusive access to technologies, and it barely sometimes slowly trickles down to the poor, less fortunate populations or even countries. And so this is a tactical question, like in an ideal world, everyone would have access to all the technology. Of course, we don't live in that world, but you can separate out how we deploy technology from should we do it at all. And I think in this case, you can introduce more mutations and increase rates of fertility, so there's more diversity in genetic selection. That's actually a good point is that why we should optimize the power of genetic technologies, because you could say, well, if there's more and more selection and people just start marrying each other's like super partners or whatever they would look like, or only people like all the blondes get together and say we're only having blonde kids, is for some people a dystopia, for some people a utopia. But how do you ensure that they're not decreasing diversity? What you can do is use the same genetic tools to increase dramatically the diversity that's present. So I think if the technology is good enough, if you want to increase mutation rates or increase general diversity, or if there's genetic markers for fertility, you could potentially do that. Now, it assumes we know them well enough to make such edits, but assuming that's all true, you could use the same technology to solve those problems. But I'm a technologist and a geneticist, so I think like technology can solve genetic problems, and of course that's it depends on how it gets deployed, how accurate it is, whether it's pleiotropy that we understand or not yet. So all these are factors that you have to consider well before you do any of it. And I'm also a big advocate of doing, if you can, multi-generational trials of any therapy, including like the Framingham Heart Study did this, and they're still doing their study, for example, which we talked about before. Yeah, but any other questions? That's look ahead.
Yeah, I just want your opinion on something. So something that Mike talked about a little bit is polygenic risk scores.
Oh yeah.
And so it's really cool that you can predict well in some diseases based on like a million SNPs, right? That a disease is gonna happen, but like what do you do with that information, right? Like it's a million SNPs, so you're not gonna target like
all. Yeah, in multiplex CRISPR, so we talked a little bit about actually in the colossal data sets we're looking at, for example, they're trying to do multiplex CRISPR for dozens, someday maybe hundreds of loci for actually de-extinction. But clinically, for example, yeah, you wouldn't, there currently is no way to multiplex CRISPR edit tens of thousands of loci, certainly not millions, right? So not even hundreds, you can maybe get to over 100 barely today. Well, let's presume that gets fixed and improved, and eventually you could do every known high penetrance allele plus all the contributing alleles, assuming there's no pleiotropy that you disrupt, which is there's almost guaranteed you disrupt something, but assuming you somehow know that that's all been solved and you've checked over many generations, that would be, you could do that actually, and then you'd have to, that would be, I think, the ultimate test of your polygenic risk score is, okay, yes, I mean, it's a good question, Nick, because if you did all that and then the disease still came, it's okay, there's something else happening. It's either something in the environment or there's some other enhancer or regulatory element that we missed. But realistically, I bet you will see that first in mouse models where someone's looking, we got a perfect mouse model. We have these 480 loci that we think contribute to the diabetes phenotype. Let's change them all back to some other allele and see if we can get rid of the risk. So I haven't seen papers on it yet, but I bet you will see that in the next 1-2 years. Is people say if we're really going to test the PRS model, let's get rid of all the alleles. But you'd have to do it with multiple generations because right now, I don't know. The record I think so far is about just over 100 for multiplex CRISPR. And there's other variations now of Cas enzymes that are more specific. So there's at least like 10 companies I know of and a bunch of research groups just trying to find more variations of Cas enzymes that have less, off target or more variations or variations of prime editing that are coming out of David Liu's lab. So I think it's it's a fascinating time because we'll see that, I think, very soon.
I didn't know they could do so much. That's interesting.
Yeah. It's crazy. I mean, a lot of these ideas have been around for, decades, but now it's no longer hypothetical. And that's the other thing is that a lot of discussion, I was just talking to a guy, there's going to be an article coming out in Wired in like a few weeks where that was the big question. The reporter was like, what if somatic editing gets so good we never even have to consider germline editing? Like, what if we just don't, because it's just so good, it's no longer really a controversy because you could just modify it. And I said, well, and that's what I told him, as I said, there's two cases where maybe you would want to, it's resource dependent or where you have no ability to have a wild type allele that you have to actually do it for both of them to have a healthy baby. But other than that, it's only two special cases I can think of actually. Yeah.
Sorry, sort of a follow-up to that. So do you think there are specific, so I know that there's like variations in like CRISPR-Cas9 delivery efficiency, and I don't know how many studies have been there about specific cell, delivery efficiency with different cell types, but do you think there are particular cell types where it would be impossible to do somatic genome editing?
Yeah, great question. So yeah, delivery is historically one of the biggest problems for drug delivery, for therapeutic editing. Some of the ones that are of less accessibility to basically just vasculature, those are less perfusion, they'll be hard to get to. The LNPs, the lipid nanoparticles used for vaccine delivery, a lot of them end up in the liver. So if you're targeting a liver problem, that's great, but for other tissues and cells, it's much a big problem. But even actually one of the things is like this map of, for example, these are all sort of the surface markers on different cell types. So one of the papers we published, we're in the middle of updating this, but some of the epitope maps of cells, we're still figuring out like what's just on the surface of most normal cells anyway. So the epitope maps are still ongoing. I think in the next 5 years, we'll probably have most of them tracked down. So then you can get combination T cells, for example, that have an if and or an or statement essentially on the surface that if we see this receptor bind, if not, then to look for a different receptor, which we detailed a bit in this paper a little bit if you want to read about sort of the concepts and first designs. But I think the delivery will get better once we finish mapping each one of these epitopes. This is for cancers, but you have to do the same thing for normal tissues. Also, but really it's going to be perfusion. If you can't get into the tissue, then you have to do like manual injection to get there if it's hard to reach. But I think, but you could start coding. You could either program the cells, other other variations of delivery system that aren't nanoparticle based, but maybe it could be something like you know, engineer the immune system to be the delivery system. You could do variations of adeno-associated vectors, but they often have multiple insertion sites and can be more problematic. But another area of direction. But that's one of the hardest questions in drug and therapeutic delivery is localization. How do you get it to the right cell in the right part of the body? And it's solved for some, but not others. And so, but I think it'll get better because we literally don't know everything on every cell type yet. And the human cell atlas, actually, we didn't have any talks on that here, but it's just an ongoing project by Aviv Regev and others. And actually, you're going to the lab in the UK, the fellowship. Whose lab? It was
Techman Lab.
yeah, the Sarah Techman's lab. It was Aviv and Sarah for the most part. Like basically said about 10 years ago, they said this very question, like, we don't really know what's on, we don't even know how many cells there are on the body, what's on their epitopes, like where they are. So the human cell is really this big outgrowth of just that pure question of what is in the body and how many cell types are there, what are they expressing, what on their surface. So so the Teichmann and Aviv's lab, but Aviv's now at Genentech. I think she's still kind of doing it at Genentech, but it's part of her labs at Broad still doing it. But it's just well, I think once we've done that mapping and some like spatial mapping technology too, we'll better delivery. So, did you have a question as well or no? What are some what are common off-target effects of Cas9 editing for blood-related disease? Common effects, some of them are for the editing of T cells you try to treat a lymphoma, but you end up getting a CAR T induced lymphoma because you can actually see it impact. We published one case report on that. So the biggest challenge is, and this happened with the early trials of gene therapy, is Jesse Eisinger was the case where one of the very first gene therapy trials that died quickly, or other people have gotten cancers, but that you run into an oncogene or you disrupt a regulatory element that's preventing an oncogene from being active, you can create cancer. But the other thing is just we don't know actually a lot of them because it's only been utilized clinically for 6-7 years, 10 at most. And you can actually see some of the first therapies. CAR T has been used of it here, but this is, you can kind of see some of the very first modified T cells were 2007 and 8 for the most part. But so it's early, so we don't know all of them yet. A lot of these weren't even CRISPR modified. These are just ones that were modified T cells. So we'll see. But if it's happening to someone when they leave their DNA behind, we can just sequence it and tell whether they've been CRISPR modified maybe in the future.
So actually, just to clarify, so the CAR T cells themselves become lymphoma.
Yeah. Which is actually, if I can find, it can. It's rare. I think it's probably, it's one of the things, probably 0.1%. Like we've only seen a handful of cases, but it's not 0.0001%. It is something if you consider you've modified a T cell and even CRISPR or done a variation of it, it may be perturbed. And so I think there's a lot of work from Prodigy is one of the automated systems to say during the cell manufacturing process, which is like any manufacturing process, how do we QC the cells and make sure that they're right? They don't release a bunch of cytokines, or they don't have other problems. Obviously, you can sequence the genome and look for off-target mutations, other insertions, ideally with long reads.